So we're going to start looking through this here. So I really want us to kind of take a look here at this image first before anything else. So we're going to look at something here called incomplete dominance. Basically, what this means is normally when I say, hey, here's a dominant phenotype, here's a dominant genotype, that means that color is going to prioritize. With incomplete dominance, that's not necessarily guaranteed. Instead, you're going to get a mix. And as an example here, if I mix a red flower and a white flower, I will get a pink flower. So that is incomplete dominance. So the best way to kind of remember this is just with this image down here. You mix a red flower and a white flower, you're going to get a pink flower. Again, big thing here, if you're really trying to think about it, is just these flowers here. And hopefully we'll be able to infer the rest of this information from that. Now this is different though. So we go from dominance to incomplete dominance to something called codominance, which where basically both of these alleles are shown. So, one example here is a red and a white flower is going to create a red and white flower. Same thing with the cows, black cow, white cow, you're going to get a spotted cow. Again, so we go from dominance to incomplete dominance to codominance. So dominance, red and white flower, you're going to get a red flower. Incomplete dominance, you're going to get a pink flower. And codominance, you're going to get a red and a white flower. So there's a couple different, so you have those different variations here, there's different combinations. And so when we look at codominance specifically, there's a couple diseases here that can be affected. One of them is sickle cell disease. You don't need to know what it is. You just need to know that people who are heterozygous for this have both normal and sickle-shaped cells. Specifically red blood cells. So normally your blood cell looks round. This is one that kind of looks like this. So these are people who are heterozygous. So they have a dominant and a recessive gene for it. The majority of Africa has this. The nice thing about it, though, is that these people who have this have a higher resistance to malaria. So it actually is an evolutionary benefit. And we'll end up talking about this, but the only real vital points here, though, are that one, this is a heterozygous disease, and that two, because these people are more likely to survive malaria, it's very lethal, it kills millions of people a year, they're more likely to live versus people who don't have this are more likely to die. They are more likely to pass that on to their offspring, which is why it is endemic at the region. It is everywhere there, and that's actually a good thing. So there's one more interesting way that dominance can kind of adjust. Now, before I go into that, does anyone know what types of what type of blood humans can have? There's A, B, there's O, and there's AB, but there's also positive and negative. So you have a bunch of different blood types, actually. And so how does that work? So you don't need to worry about drawing this. Don't worry about that. But so you have 4 different blood types here. And where these come from are from the various combinations of that you can get this. The big thing here, though, is that blood is specifically addressed by 2 alleles. So we have something called multiple alleles now. So there's multiple forms of dominance. So we have A, B, and O to form our blood groups, but now we can force them 3 different ways. So you can have A, this is AB. Yeah, so there's a whole bunch of different ways. You can also have positive or negative associations with it as well. So what we call all of this, and we call these the AB markers. And there have been lots of incidents where people have discovered that, one of their parents, for example, was not, couldn't have been their dad or their mom, just based off the fact that they figured out their blood type. And it's like, that doesn't work. I can't have a double B, but both my parents are A, for example. So this is just an enlargement of that. We'll end up coming back to the blood stuff later and we'll make a little bit more sense of it. But so these multiple alleles, this is still multiple alleles. One example here is with the color-coded rabbits. Yeah, so here we go. So yeah, so if we're looking at this, I quickly want to go to this. So you can have a priority in how it's operated. So black bunnies are full color and then not full color, then Himalayan, and then albino. And we're going to talk specifically about this part here soon as well. But so we can have a hierarchy of dominance, a priority list. So one example here is in rabbits, we have 4 different alleles that code for coat color. In human blood, it was 2. Most of the stuff we've looked at has been 1. Now we have 4. So we have a hierarchy of dominance here. So basically what this means is if this is present, then this takes priority over the CH. If CH is present, it takes priority over just the H, and so on and so forth. Okay, so again, just kind of looking at this here again, so you can have a hierarchy. These are the 4 possible alleles. Again, there's 4 different possible ones. Where this differs is you can have uppercase, lowercase, you can have the CH, the H, or just the C. So there's 4 different ones. Whereas normally we're used to, maybe something like this, dominant recessive, for example. We see this a lot in dogs as well. So this is useful information if anyone's ever looking to be a dog breeder. So if we're trying to breed dogs, for example, these are the genotypes of this. There's no dark pigment. But the second I introduce a dominant E, this is going to affect the darkness of it. So where this comes from is something called, we call this epistasis. So this is when one allele hides the effects of another allele. So I'm going to just go back to this picture here. But basically, when we look at these Labradors, if we have the dominant E, this chooses whether the coat is going to be a dark pigment or not. And then the second allele chooses how dark it is. So the E's determine if it's dark, yes or no, and the B's determine the color itself. So E determines whether it's dark, B determines how dark. If it has multiple recessive alleles, it's going to be yellow. And that's because it masks the effect of the B. Basically, even if I have a dominant B, because I've said, hey, these E's here are recessive, that means it cannot have a dark coat. Even if it was meant to have a black coat, it can't. The fur doesn't allow it. And then one other variable here is looking at sex determination. The one thing to note is when it comes to, you know, everyone's probably seen like videos of this or like heard about it, but, oh, I have like, you know, 4 sons and I really want a daughter, or I have 4 daughters and I really want a son. You probably know someone who's, you know, said something like that. The person who determines the gender is actually the male. It comes from the male's side. So the male side is what determines the actual gender. So we have sex chromosomes. We know that XX is female and XY are male. And then the other 22 pairs of chromosomes are called autosomes. So if you recall, you have a total of 46 chromosomes. 2 of those are dedicated to just the gender. That's it. The rest of them deal with the other stuff. We are going to actually look at those 22 chromosomes later. The one thing I really want to note here is, actually, I'm going to blow this picture up. Give me 1 second. I want us to kind of clue into this picture here. This one, okay. Nope, not that one. This one. So I'm just blowing up this picture here. But again, so so this is a Plunkett chart of, we have XX, the mother, XY, the father. So one thing to note here is that the sons only get their Y chromosome from their father. It's the only place they can get it. And so if this Y chromosome carries something, a disease, for example, color blindness, which it does often, the males are immensely more likely to have that. Or so one of the things is if it comes from the Y chromosome, that can disproportionately affect males if it's a dominant gene. The other one is, what if my mother has a dominant thing over here that only affects the X chromosome? If that's the case, and they have a son, they have no other X chromosome to stop that. Or sorry, maybe they have a recessive gene. So maybe they carry a recessive gene that causes, I don't know, a much higher rate of cancer, for example. They have no other X gene that could have a dominant gene to kind of supersede that. So recessive genes on the X side, much more common with males. And dominant genes on the Y side, much more common with males. And the mother can give you either one of her X chromosomes. But if we have a daughter, the X chromosome can only come from this one. So a lot of the times when you're looking at females, your mother's family history matters a lot more. When you're looking at males, your father's family history matters a lot more. So males are just genetically more predispositioned to being more likely to get certain medical things. Again, like one of them is color blindness. It's like 10 times more likely than females. Because for a female to get color blindness, if we assume it's recessive, it has to come from both the mother and the father, and both of them have to have that colorblind gene. So this is a complicating factor with it. And so that's a complicating factor with those pedigrees, for example. We'll end up looking at some examples later as to, looking at family histories and trying to conclude which side of the family did it come from. So this X chromosome carries a bunch of those genes that are, the X chromosome carries genes that are necessary for the development of both females and males. Because it doesn't matter what you do, you will have an X chromosome. You will have it. The difference is the Y chromosome has genes that relate to the development of male characteristics specifically. So specifically male characteristics. And in females, one of those X chromosomes is inactivated at each cell. There are two of them, right? So they're going to shut one of them off. So we call this dosage compensation. Another word is X inactivation. And they have used both of these before. The one thing to note, though, is that whichever X chromosome between these two stops working, purely random. I'll stop going back and forth now. So one example of this that we see with specifically this X activation. So when we have a female and they both have a gene for the same thing, maybe it's a gene for hair color, eye color for humans. Maybe it's eye color or the coat color or fur type in animals. One of them is going to stop working, and it's purely random. The question is, sometimes that might affect what the animal is going to look like. One example we see of this is a calico cat. So if you guys are looking at all the black parts of this cat, so this is a female. What happened here is the gene that was responsible for that, it turned off. So like maybe this was supposed to be orange or white or a brown or a different color, but because it got deactivated, it just came out as black, for example. That's just how like the pigments that's just how the pigments are and how visible light hits our eyes and so on and so forth. So this is an example of where this activation happens, and it actually does have an effect. This is why a lot of calico cats look completely unique, is because one of the genes gets turned off, and then that gene portrays as black fur. So again, just as an example of this, what this looks like here, we have a gene here. This is our allele for orange fur. This is our allele for black fur. As it gets split, one of these gets deactivated, and the other one is activated. I don't think we need this. No, we're good. We don't need that. There's also sex-linked traits. So these are traits that are controlled by genes located on the X chromosome, and they are sex-linked. Again, this is what I was talking about earlier. Because males only have one copy of the X chromosome, they are more affected by recessive X-linked traits. Okay, so if we're looking at color blindness specifically here, again, so color blindness is an X chromosome trait, and it is a recessive trait. So with females here, for example, if I have this is the male's genes, right? Because it has the Y chromosome. If the male has is not colorblind, if I have a kid, I have a daughter, and I myself am not colorblind, that daughter will not be colorblind. It's not possible, because no matter what, they're always going to have a dominant gene, unless the mother is also colorblind. So mothers are carriers, right? That means that. So simply put, males are more disproportionately affected by color blindness, because if you have a mother who is a carrier, you have a 50-50 shot if you're a boy, being colorblind yourself. Whereas for a daughter to be colorblind, both parents need to pass on that gene. The man has to be colorblind, and the female has to be a carrier or colorblind herself. So when we're looking at recessive X-linked traits, it really matters on your mother's side. Males are more disproportionately affected. So one example of this, I'm going to blow this up. This is of Queen Victoria in England. So this is their family's history and their pedigree. And this is specifically looking at an X-linked trait that causes clotting of blood. So a recessive X-linked trait. So if we remember, this Queen Victoria herself was a carrier of it. But if we're looking at who is actually affected here, it is only males. It's only males because they are getting, again, that 50-50 shot. They are getting with they're an offspring of a carrier, and then that means that they themselves have a 50-50 chance of carrying that recessive disorder. So the second we have a carrier involved, it is a 50-50 shot whether that man is going to have that or not. And we're almost done. Almost done. We also have things called polygenic traits. So these are when we have multiple pairs of genes. So these do affect things like skin color, height, and eye color. Again, if you have two tall people who have children, there's no guarantee that they're going to have tall kids. It is more likely, yes, but it is not a guarantee. And a lot of that has to do with the number of dominant alleles in there. For example, the chart you can see here is skin color. The more dominant alleles there are 3 of them, A, B, and C's. There's 3 different alleles that affect it, multiple genes that affect it, the darker the skin color. So I really want to make a note here. This is multiple pairs of genes, not alleles. So there are different genes that affect skin color. So there's one more factor as well, and that is our environment. There are more than just these three things, but these are common examples. So your environment can cause genes to portray differently. So with sunlight and water, for example, we know plants won't produce flowers. If there's not enough water, plants start to lose their leaves. Pine cones will sit there dormant for years waiting for the right conditions. So will other seeds, etc. The other thing is that lots of animals specifically, they change what they look like, the phenotypic, what they look like with extreme heat or cold. We see a lot of examples of how temperature affects organisms. Bunnies, for example, a lot of them end up turning white in winter. We see cats, for example, as well, in warmer climates, colder climates, white cats will exhibit more black features because it absorbs sunlight a little bit better, right? You think about like when you get cold, where do you lose most of your heat? Your extremities, your head, your feet, your hands. Oh, sorry, your head, feet, and hands. It's the same thing for cats, right? Their nose, their head, their feet, their tail. That's where they lose a lot of their heat. And so by changing that to be a darker color in colder climates, they're able to absorb more sunlight. And then this is another example of environmental factors. How can we, if we look at people live very complicated lives, they all grew up in different circumstances. How can I determine, for example, let's say I am studying alcoholism. Is there a genetic disposition towards alcoholism? If so, how much? That's a hard question because I can't just compare 2 different people because they grew up differently. They had different lives. They played different sports. Maybe, they had different friends, different interests. They have different genes. It's very hard to do, which brings us to twins, specifically identical twins. If we compare identical twins who have the exact same DNA versus fraternal twins who do not have the exact same DNA. And if we look at the rates of certain things, we can then determine whether something's genetic or whether it's environmental. Again, if we're looking at let's look at depression here, for example. There's a little bit of there's a lot of genetics that actually are in play there. Versus fraternal, a lot of that seems to be environmental. So when we look at twins versus non-twins, this helps us figure out what is genetic and what is environmental. So if there are traits that happen a lot in identical twins, so if both twins develop, for example, really high blood pressure, that probably means that it's controlled by heredity. So that probably means they inherited something that influences their blood pressure. Whereas when these traits are expressed differently, so when they are expressed differently, that's when it's more strongly influenced by the environment. Again, you look at the blood type, your blood type of twins is completely genetic. We look at reading disabilities, it's really high, right? Over 60% of identical twins, if one has a reading disability, the other one has a reading disability. Why is that? A lot of it's genetics. If we look at something like alcoholism in females, though, that does not seem to have a lot of genetic factors. It has a little bit. We do know that alcoholism is partly genetic, but it's not a lot. So that is our advanced stuff here. We will look at a little bit additional complexities probably tomorrow. But I quickly want to go over 3 different questions here beforehand. You might want to. Yes, I do. Make this your 3rd question. This is the big one. This is the big one. The other ones, you really just need like 1 to 2 lines for the other ones. So move that genotypes of the parents, the blood types, to the bottom, please. Because that's the big one. Okay, so I'm going to start going through some of this. So epistasis and dominance, how are they different? If we recall, epistasis, where did I put that in here? So epistasis is where one allele hides the effects of the other one. So one allele hides the effects of the other one. How is this different than dominance, right? Doesn't it seem that this E is dominant to these B's? Well, with epistasis, it masks the other one, right? So what this means is a recessive allele can mask a different dominant one. Whereas with dominance, whatever's dominant reigns on top. But now what happened, as we saw at the Labradors, is recessive genes can overwhelm the dominant gene. It can mask it. So the main difference between epistasis and dominance is that epistasis allows those recessive alleles to control it. Or sorry, rather to take priority. Okay. Coming down later. Twins. So how are twins or clones? That's starting to happen with animals. How is that important with differentiating the effects between genetic and environmental influences? This matters both for animals, but also humans. We look at different things, but basically we know that identical twins are genetic copies. So when we have traits that portray identical and traits that portray differently, that must mean that those come from the environment. That's poorly worded. Give me 1 second. Let me edit that. Traits that portray identically and traits that portray differently must sorry, traits that portray identically come from genetic influences. Traits that portray differently must come from the environment. Better, better. Okay, and then finally, these genotypes with blood types. So again, if we have blood, so we have a father who's blood type A, we have a mother who's type B, we have a daughter who's type O, a son who's AB, and another son is B. And we want to try and figure out what are the genotypes of the parents. So to do this, I want to go back to that blood stuff we were talking. So this is this chart here, right? So this is showing all the possible combinations that we can get. Or put a different way, these are our blood types. And I want you to write just the chart down, this the thing I'm highlighted in red. So these are the possible genotypes. Again, your blood is determined by two things, which means you can have type A blood with either one of these. So knowing this, so if we know the possible combinations, I want to go back just 1 second here. But yes. Oh, actually, no, I want to go here. So I can only get an O type blood if both parents pass along their recessive gene. So knowing this, I know that the parents have to be heterozygous. So that's one thing. That's a clue I have. So I know they have to be heterozygous, which means they have to have one of these two things. I'm sorry, I did a mistake here. If you look on your B, this is supposed to be a B. Replace that A with a B. I apologize. So again, I can only get O type from having a recessive gene from both parents, which means both these parents have to carry that. So that is my clue number 1. My second clue here is that I know the other kids we have A, B, and we have B, which means that they carry both an A and a B. So they have to have these two genes here. So if we know that we have a son with O type, that means they cannot have this. For one, and then we know that they have ones with A and B, which means it cannot be this either. So just by deduction, we can get this as their phenotypes. We don't know which one's which, but we don't need to know which one's which. So these are some examples, again, how you can determine like blood type. You can, and people have been able to look at this before and be like, oh, like this shouldn't be possible. If the kid, for example, if let's say the mother's blood type, for example, was just A, it was just blood type A, and if they had a kid with O type, the mom cheated. It's a fact. She had to have cheated. Or there's an incredibly rare genetic mutation that's happened. Those are the only two options. So you can use this even without a DNA test to determine results of parentage, for example. Okay, so that's 2 o'clock. Let's take a break here. Take a 15-minute break. We'll give you guys 20 minutes, actually. That was a lot of writing. Give you 20 minutes. We'll come back in 20 minutes. So just to kind of finish off here. Ooh, here we go. So just finish off. So today you'll be able to finish up, if you haven't yet, 10.2. Oh, sorry. Oh, sorry, not 10, 11. You'll just be able to kind of finish up those reading parts here. Again, that essentials is not 4 marks. Tomorrow we will be finished this, and we're going to start with a fresh, clean slate on Monday with Unit 12. So this is kind of a plan here for the rest for today and tomorrow.